Hello students, welcome to Vedantu NEET English. I'm Ms. Gopika, your own biology master teacher. I hope I'm audible and visible to all of you. Please let me know in the chats if I'm audible and visible. And welcome all of you to yet another amazing session where we are going to do biological classification part 2. So last class we had done the introduction of the entire biological classification where we learned about the kingdoms, we learned about the drawbacks of each kingdoms, and we have also learned about kingdom Monera. Now we are left with kingdom Protista, fungi, virus, viroids and lichens. Kingdom Plantae and kingdom Animalia will be taken up individually as plant kingdom and animal kingdom. Okay, so please let me know if I'm audible, visible to all of you, and quickly share this video with all your friends who are lazy and who are not consistent. Just by watching part 1, you're not going to be able to answer all the questions, right? You have to be consistent. You have to answer part 2 as well. Okay, yes. Yes. Am I audible, visible? Did you all mark your attendance? How will you mark your attendance? By smashing the like button. Okay. Yes. Thank you so much. Good evening. Good evening. Okay. So all of you are ready? Let's go. Let's start. Okay. Without glasses, no vision. Okay, great. So students, we have to start with Protista. Today we have a lot to cover, but we will try to cover as much as possible. No pressure. We have enough time, so we will take it slow. Okay, we will take it slow. Yes, please give me a fire emoji if everything is proper. Okay, quickly give me fire emojis. I've told you last class also. If you are enjoying the class, if you're understanding the concept, you keep putting fire emojis. The chat has to keep moving. There's no way that the chat has to stop. Okay, one person put, the other person gets motivated to continue listening, and it goes like a cycle. Okay, so students, for those who do not know me and watching me for the first time, I am Gopika and I have done my post graduation in zoology, and that is why majorly I take zoology classes, even though now we are watching a botany class. Okay, and I have worked as an assistant lecturer. I have also done my BSc CBZ, that is chemistry, botany and zoology. Okay, this is about me. So yes, I have been working here in the online mode from past 2.5 years. Okay, and offline separate. So students, some of you were asking me, ma'am, especially students from Bangalore, there is a JP Nagar center for all of you. I will give you the details of the center in the end of the session, so just stay tuned. Okay, if any of you have any doubt, be it for JE and NEET, we will be covering it in the end of the session. Okay, chalo, let's start. Awesome. Good evening all of you. So students, today we are going to start with kingdom Protista. So last time's notes I could not download. There was an issue with the download option. So today I will try to merge both the PDF and share it on the Telegram channel, Vedantu NEET English Telegram channel. And if you want to join my Telegram channel, just put my name and my subject and you will get my Telegram channel. Okay, so yes, please be part of our Telegram channels. So students, starting with kingdom Protista. What do you know about kingdom Protista, students? Whatever you know, please keep answering. Okay, so when we spoke about Monera, we had a clarity that we are talking about a unicellular group of prokaryotic organism. But when we slowly move to Protista, we understand that we are basically talking about a group of unicellular eukaryotic organisms. Okay, eukaryotic organisms, right? So when I say eukaryotic organism, there is a sentence given in your NCERT that talks about these people have membrane bound organelles. So I can just say that eukaryotes means, yeah, they are going to have membrane bound organelles, but on the prokaryotic side, they're not going to have, right? So here one more statement comes into existence, students, that these people have membrane bound organelles. Okay, membrane bound organelles. Done? Yes. Now there's a problem with Protista. Some of you might relate to this. Some of you do not have boundaries. What do I mean by boundaries? Some of you do not know when to tell your friends no. Some of you do not know when to tell your parents no. Some of you do not know when to tell no to strangers. You do not have boundaries. So when you do not have boundaries, that means what? Anyone can come into your life, move out of your life, come into your life, move out of your life. They can make a khichdi out of your life, and you are not going to have a problem with that because you do not know how to say no. At the same time, there's another set of people who do not have boundaries. That is, they don't know when to stop. right? When to be respectful, when to be, you know, fun, that kind of people. So something like that, Protista also, this entire kingdom does not have boundaries. That means for some people who are only studying about Protista, some of the organisms are Protista. But a person who is a proper botanist and let's say he's only focused on plant kingdom, he'll be like, no, wait, I want to put these people in plant kingdom. So such kind of confusion happened between Chlamydomonas, Chlorella and Spirogyra. So some people studied individually, some people studied under Protista. So it is people to people. That is why I told you these people do not have boundaries, right? These people do not have boundaries. So students, here something similar happens in Protista also. These people do not have proper boundaries. Okay? No proper boundaries. Okay? Now, in kingdom Protista, okay, when I talk about kingdom Protista, you need to understand that kingdom Protista has again divisions. Okay? Who can tell me the divisions quickly in the chat box? Come on, all of you. Yes, nucleus is present. Very good. Okay? This we know that nucleus is present. Now, Protista is further divided, that is, they have again classification where you have few people who are similar to plants, few people who are similar to animals, few people who are similar to fungi. So, we can say that Protista is a link between, okay, it's a link between plants, animals and fungi. Can I say that? Yes, ma'am, whatever you want, you tell, ma'am. Is that your attitude? Very good. Chrysophytes, okay? Chrysophytes, dinoflagellates, okay, I think we'll have to extend the arrow mark a little. Okay, what do we have here, students? Uglyoid. Here, what do we have? Come on, quickly in the chats. After Uglyoid. Okay, I think you people are already giving the examples. Okay, just tell me. Yes, very good. Slime mold. Okay. Slime mold, and in the last we have protozoans, right? We have protozoans. Now, what is the so special about these people, ma'am? What is so special about these people? Let me break it down for you, students. The thing is, chrysophytes, phytes. Somewhere we talk about plants, right? Phytes, words. So, these chrysophytes are something who can make their own food. Some relation is there with plant. Again comes dinoflagellates. Again, the similar thing is observed. Dinoflagellates as well. So, if you notice, these two people, right? These two people are going to contribute, or they are plant-like. So, I told you Protista is a link between plants, animals and fungi. So, these two are kind of plant-like, okay? Now, comes Uglyoid. Students, Uglyoid is more of a mixotropic, okay? That means it has some plant-like characters, it has some animal-like characters. So, it is more of a mixotropic, okay? And when we come to slime mold, mold means we always talk about fungi. Right, so here comes fungi and protozoans. Okay, zoans, again zoology, so that comes with animals. Okay, you can remember like this, or you can remember normally also animal like. Done? Understood? So can I say Protista is a link between plants, animals and fungi? Yes, ma'am. Yes, ma'am. So I would write here Protista is a link between plants, fungi and animals. Done? Done. Yes, all of you agreed this? Yes, correct? Okay, shall we go further? Okay, shall we go further? Okay, so kingdom Protista, we have chrysophytes, dinoflagellates, uglyoids, slime molds, and again, protozoan is further divided into amoeboid, flagellated, ciliated, and sporozoans. Okay, all these names you are aware. It's not like first time you're hearing this name. You know amoeboid, you know ciliated, you know flagellated. So these terminologies are very aware for us. Okay, Now, we will start with the first one, students, chrysophytes. So, whatever you know about chrysophytes, keep, putting it in the chat box. I can already see some of you are already put. So, students, chrysophytes, the first thing is they have two people coming under them. Who are they? Diatoms and desmids. Okay? Diatoms and desmids are again called as golden algae. See, we are using the term algae. Okay, we are using the term what? Algae, right? Students, I might write a little like, slanting like flight takeoff, because I'm very used to writing like that. Okay, now these people are present both in fresh water. Okay, they are present both in fresh water and marine. Okay, all these lines are from NCERT, so you do not have to worry. Now, students, when we studied R.H. Whittaker's 5 kingdom classification, we learned that when we talk about kingdom Protista, few of them have cell wall, few of them do not have cell wall. But let me tell you, the most amazing character of this kingdom Protista is every cell wall is specific, every cell wall is peculiar. Now, why am I telling that? Because here, when we talk about the cell wall, you should remember that these people's cell wall also is nothing out of, it is not something normal. It is like special, because this cell wall is made up of silica, and silica is not easily destructible. Okay, for all of your information, it's not easily destructible. And this silica, this cell wall is there, no? It's made up of two overlapping shells. Okay, one second. Two overlapping shells like this. Like a soap box. Like what, students? Like a soap box. Something like this. Clear? Like a soap box. Can you all tell? Yes or no? Takshi. Very nice. Okay, so this is what is happening. It is like a soap box, like overlapping shells. Can you all remember this? Yes or no? Students, all of you go to your washroom and take a soap box and come and see how it will be looking like. One will be like this, one will be like this. Okay, in between this, you can keep a soap. Have you seen that before? Our parents used to have that card holder. Even the card holder used to be like that. The bottom used to be a little larger, the cap used to be a little smaller. So same thing here also. This silica is formed into an overlapping shells. Okay, I hope all of you are marking your attendance as well. Yes, great. Okay. So, these people have two overlapping shells. Okay. Two overlapping shells which are into a soap box like. Okay. Now, I told you in the starting that these people are plant-like, but here nowhere I am talking much about it. Yes, I did talk about the cell wall. So here, students, remember, most of them are photosynthetic. Okay, most of them here are photosynthetic. So when they are photosynthetic, where will I place them? That means their mode of nutrition is going to be autotrophic, right? So if that is the case, I'm definitely going to place them like plant-like. You are very similar to plant-like. So same thing I'm telling here. Most of them are photosynthetic. Now, one more most specialized characteristic is that, students, this overlapping shells which are made up of silica, no? These people, what they do, they go to the bottom of the earth. Okay, let's just consider like this is the earth. Okay, this is the earth. These people go and accumulate after the organism die. These people will go and accumulate at the bottom. Okay, bottom of the sea. Like this, and they form something called as the diatomaceous earth. What do they form? The diatomaceous earth. Diatomaceous earth. Now, students, this diatomaceous earth, what will be the characteristics? Ma'am, it is very simple. This diatomaceous earth is going to be silica rich, right? It is going to be rich in silica. And second most thing is, students, these people are deposited over a long time. It's not like today one shell falls and it is like, oh, diatomaceous earth is formed. It's not like that. It is a very long process. Okay, it's a very long process, right? So that diatomaceous earth is going to be silica rich. Yes, ma'am, it is going to be silica rich. Correct? Did I write an A by mistake? 1 second, students. Yeah. Okay, silica rich. Second thing is, students, I told you silica is indestructible. That means you cannot easily destroy silica. So these are also going to be indestructible. Yes, ma'am. Third thing, students, what would be the use of this diatomaceous earth? So they have like they are like gritting. Okay, that is like, you know, sharp. Have you seen sandpaper? How many of you have seen sandpaper? Raise your hands quickly. Yes, Advika has put a target. Very nice. Let me also see how many of you are there in the class. Okay, 1 second. There's no lag, no? Okay, cool. Yes, quickly mark the attendance. We are almost there. Okay, yes. So students, all of you have seen sandpaper? Yes, most of you have seen sandpaper. Okay, very nice. So students, that is like the gritting. That means it can be used for polishing. So here this diatomaceous earth, we are going to use it for what? We are going to use it for polishing. And between this silica, there is going to be spaces which is going to allow us to do filtration as well. So if they ask you what are the functions, okay, you can write polishing and you can write filtration. Clear? You can write polishing and you can write filtration. So that is the job of your diatomaceous earth. Okay, I'll write this down. Soap like. Soap box like. Okay, soap box like. Any doubts so far? Just let me know if you have any doubts here, please, quickly. Yes, it is going to be very rough. Okay, very rough, and they are going to help in what, students? They are going to help in filtering and they are also going to help in polishing. Okay, filtering and polishing. Clear? Yes. Perfect, perfect. So this is clear, students. So the cell wall here is going to be overlapping, and this overlapping is going to look like a soap box like pattern. Okay, and this is made up of silica, which is indestructible. Over the years, they are going to settle at the bottom of the sea and they are going to form diatomaceous earth. That is taken up by the humans. They use it for polishing as well as for filtration. Clear? Polishing and filtration. Clear? Okay, just give me a thumbs up if it's clear for all of you. Then we can go to dinoflagellates. So students, dinoflagellates. First, let's just decode the word. Okay, what is the meaning of it? Dino means die. Okay, here, what is the meaning? Two. Right, two. Flagellated means flagella. Right, flagella. So two flagellas, right? This is what we have understood from the word. It's not that difficult to, think of it. Yes. Now we will go into the characteristics. Okay. These people are, students, mostly marine. Mostly marine. Now, one very special character about these people is that these people are present in different colors. Okay, they are present depending on the pigment that they have. Depending on the pigment that they have, they are present in different colors. Okay, so they can be seen in green. Okay, they can be seen green color. They can be seen in yellow color, red, and you have to learn more about red. Okay, then which color, students? Brown. We do not have brown here, so we are going to use something like this, like an elephant grey. Okay, doesn't look like brown, no? Will you people get doubt? Ma'am, brown, ma'am. Okay, yellow, green, brown. Okay, one more color we have, that is going to be blue. Okay, that's going to be blue. So here we understand that these people are present in different colors. What are the colors that they have? They are present in green. Okay, they are present in yellow. They are present red, and we are going to learn more about red, brown and blue. Okay, they are present in different colors, students, depending on what pigment they have. Okay, now when we talk about dinoflagellates, please understand, we again talk about the word dinoflagellates, two flagellas, right? So their structure somewhat looks like this. Okay, their structure looks somewhat like this. Okay, like this. Now, the most important characteristics, okay, they are more round here. Yeah, the most important characteristics about them is that they have two flagellas. So, students, remember that one flagella will be running transverse, okay, and one flagella will be running longitudinal. So, this is the most specialized character of this person, okay. You can either draw it like this or you can draw it like this. Okay, 1 second. I'll draw it a little more thick for all of you. So one will be going through the furrow like this, Okay, and one can go like this up. Like this. Okay, so they have two flagellas. That is the most specialized characteristics. Okay, so I'm writing it here. They have one flagella. Okay, they have one flagella which is running transverse. One flagella. Okay, let me just write flagella. Okay. Since I'm writing the notes so neatly, so you all can use it till the end. Okay, that is why I'm writing it so specifically. You'll be thinking, ma'am, why are you using this much color? Just write it in white. But it will not work like that. Okay, one flagella that runs transverse. Okay, one flagella that runs transverse and one flagella that runs longitudinal. Clear? These are the two flagellas, and that is all about their characteristics, right? These people are dinoflagellate, so that means they have two flagellas. Now students, remember, these flagellas are coming out of a small, okay, like a furrow, okay? They're coming out of a furrow, like you can see, you know, like this furrow. Now, I have seen in some of the books, okay, when you talk about this shells, overlapping shells, you will see the word hypotheca, epitheca. Just understand, hypo is the down one, epi is the top one. Okay, in case you see that word somewhere and you'll be like, ma'am, is it necessary? It's not necessary. It's not there in NCERT, but in some books it will be mentioned. So hypo is the down one, epi is the top one. Okay, done students? Is it clear? Can I see some energy? What is wrong with all of you? Come on, put some energy and just tell me, yes ma'am, I understood. Huh? Yes ma'am, I understood. If you did not understand, tell me that I did not understand, ma'am. Yes or no? Okay, wait. I think, students, am I stuck by any chance? Or are you all able to hear? Why can't I see the chats moving? Please let me know, because it's raining here. No, I doubt if there is an internet issue. Just let me know. That's why I'm asking you in the middle. Okay, it's raining here heavily. So that's why. Okay, no issues, right? Okay, perfect. Yeah, now I can see your chats. It was not moving. I got scared. I was like, what's happening? Okay, so you understood this, I hope. Now, students, when we talk about the cell wall, okay, about dinoflagellates, remember that the cell wall of dinoflagellates, okay, the cell wall of dinoflagellates is made up of stiff, okay, made up of stiff cellulosic plates. Okay, it is made up of stiff cellulosic plates. So remember, in Protista, everyone has a different type of cell wall. Some might not have a cell wall. They will have a protein rich flexible layer. Some people have cell wall, one will be different, another one will be different. So here the cell wall is going to be stiff cellulosic plates. Okay, stiff cellulosic plates. Now students, one more most specialized character. When I spoke about the red one, I told you there is a dinoflagellate called Goniolax. Okay, Goniolax. So this dinoflagellate Goniolax, student, this fellow Goniolax, he is the reason why we see red tide in the sea. What do we see? Red tide. So let's say normally this is how the water will be, no? Okay, I don't know how to draw water. Okay, wait. Okay, this is the beach water, not beach, sea water. Okay, usually it will be very calm. Now let's take example. You will notice that suddenly there is an entire red color pigment that you're seeing. Okay, and it will look like a red tide. Okay, it will look like a red tide. What is the reason for this? This is because of rapid multiplication of Goniolax. So Goniolax undergoes rapid multiplication, leading to the formation of your red tide. Now students, ma'am, is there a problem with that? It's something very beautiful, right? It's something very beautiful phenomena. Is there a problem with that? Yes, it is. There is a problem with that. Because of this rapid multiplication of Goniolax, the other organisms in the sea will get harmed because of the release of a toxin. Because of a release of a toxin called as saxitoxin. Okay. Now students, this toxin, if this toxin, see, almost all the organism will get affected because of this toxin. And now let's say we consume any of the organism who has been, you know, affected by this toxin, even we are going to end up getting a disease. Okay, we are also going to end up getting disease. The disease, basically what happens is these people are going to block our sodium channels, and that will lead to paralysis and death. Okay, so that is how they are going to affect our body. Let's say that's not me. Okay, let's say someone has consumed any kind of a fish, okay, which is affected by this toxin. Now, since that fish has entered into our body, what is it going to do? This saxitoxin is going to block our sodium channels, and because of that, we are going to end up having paralysis and death. Okay, so this Uglyoids, which is red in color, causes red tide because of rapid multiplication, leading to also a release of a toxin called as saxitoxin, which kills other organism in the sea. Clear? Yes or no? Okay, so this is about your dinoflagellates. I hope there's no doubts about your dinoflagellates. Everything is super duper clear. Okay, can we go to Uglyoids? Yes, ma'am, we can go to Uglyoids. Okay, perfect. Yes. Now, let's go forward. Okay, students, if in case I miss some points, please tell me. Okay, ma'am, this one point is there. Okay, now look here. Uglyoids. This fellow, no, students, let's take an example. Okay, Uglyoid and his friends, he called everyone. Okay, he was like, guys, let's have a party. So every friends are all those tiny organisms were like, yes, I'm ready for the party. I haven't partied for so long. Let's come, let's party. So the sun was there, Uglyoids is partying, he's having his own food because he can do autotrophism. Suddenly, there's no sun, right? It's getting dark and dark. What will Uglyoid do? Uglyoid will start eating all the guests that he called. That is, all the smaller organism it is going to consume. So that means Uglyoid have mixotropic nutrition. That is, if in the presence of sunlight, they can make their own food, and in the absence of sunlight, they become heterotrophic. So just imagine, okay? Just imagine foodie. Yeah, Uglyoid is a foodie. So Uglyoid students, the most important thing about these people are they are mixotropic nutrition. Mixotropic nutrition. It's like having cannibals, no, around us, like in movies and all we see, no? It's like that. Having cannibalism around us. Okay, so students, here these people are going to be present in fresh water, stagnant water. Okay, fresh water, stagnant water. Now let's talk more about their mixotropic nutrition. That is, students, mixotropic nutrition in the presence of sunlight. Okay, in the present, all these notes will be available for you, so don't worry. Okay, in the presence of sunlight, these people can make their own food. That means they can be autotrophic. Okay, in the presence of sunlight. Okay, should I write here presence of sunlight? Okay, presence of sunlight, they can become autotrophic. Okay, now scenario number 2, in the absence of sunlight. In the absence of sunlight, what are they going to do? In the absence of sunlight, these people are going to become heterotrophic. Heterotrophic, that is, they are going to feed on smaller organisms. Okay, they are going to feed on smaller organisms. Smaller organism. Done. Just imagine, students, we go to the party and after some time, no more guests is there because everyone got consumed. So that is what happens here. Okay, it's a mixotropic nutrition. Yes, it eats its guest. Exactly. Very good. It eats its guest. Okay. Now, students, remember this. I told you that some of them might have cell wall, some of them might not have cell wall. So this fellow does not have cell wall. Okay, no cell wall. But instead, okay, they have, okay, they have protein. They have protein rich layer, flexible layer. Okay, I'll write flexible in the bracket. flexible layer which is called the pellicle. What is it called? Pellicle. 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 So instead of a cell wall, they have a protein-rich flexible layer which is called the pellicle. So far what and all did we study? We studied that we have an overlapping soap-like structure which is made up of silica in chrysophytes. Then dinoflagellates, the cell wall is made up of stiff cellulosic plate. And when we come to uglyoids, we notice that there is no cell wall, but they have a protein-rich layer called the pellicle. Done, done, done. Yes, protein-rich layer in the absence of cell wall. Okay, I hope all of you are able to see. That's why I'm moving to the side. Okay. The board, okay. Okay, what? It's going to do the flexibility, okay? It's going to help in flexible movement, protection, okay? Mainly for body's flexibility, okay? Now, the next important thing about euglenoid is that they have two flagellas, okay? Next most important thing is they have flagella, okay? They have flagella. And here, students, there we saw they are transverse, okay? One was transverse and one was longitudinal. Here we notice one is short. Okay, one is short and one is long. So these people have two flagellas for movement. Done, 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 done. Yes? Okay. So it is for survival, no? It is for survival. Shipali. It helps them to survive and it also helps them to be, you know, prevalent in the system and in the entire ecosystem. Okay, that in the absence of sunlight and in the presence of sunlight, I will do my job. Okay, now students, remember one more most important thing about these people is they have a presence of, shall I write it? Yeah, they have the pigments. Okay, they have pigments similar to that of higher plants. Okay, they have pigments that is similar to higher plants. Similar to higher plants. This is important. Okay, this is important. Done? Got it. Okay, great. Okay, this is all about your euglenoids. Okay, next is slime mold. Now I told you slime mold means what? They are going to behave like a fungi. That means favorable condition, unfavorable condition, saprophytic, all these kind of natures can be seen. Okay, now see, they do not have chlorophyll. This is understood by us that they do not have chlorophyll. Okay, they do not have chlorophyll. Now they are surrounded by plasma membrane only. However, their pores have cellulose wall. Now students, this is one of the specialized character these people have, that is their spores are going to have cell wall. Okay, the spores are going to have cell wall, but in the previous times, that is they are surrounded by a plasma membrane. So the adult, the matured one will not have, but the spores will have cell wall. Why? As a protection, so that it can be transferred to more places. So for protection, that is given to them. Okay, now students, these people are going to have saprophytic nutrition, no doubt in that. Slime mold resemble both protozoa and a true fungi. They are like protozoa in the amoeboid plasmodial stage. That is because I'll tell you about the stage. Okay, they are similar to fungi in spore formation. This much you remember. Okay, this much you remember. They are similar to fungi in spore formation. Okay, spore formation. Now, this is how a plasmodium looks. In your NCERT, the word is plasmodium. That means aggregate, right? Aggregate. So students, remember, like this, many of them, they will come together, okay, come together in favorable condition and they will form something called as a plasmodium. Okay, what do they form? Something called as a plasmodium. This much only you have to remember. I'll tell you. See, so when the condition is favorable, okay, when the condition was favorable, now let's say COVID cases are increasing day by day. When the condition is favorable, we all go for parties, we all go for get-togethers, we all come together. But when the condition becomes unfavorable, what do? We just stay silent, we stay inside our home. So here also, when the conditions is favorable, these people are going to form plasmodium, and when the conditions become unfavorable, they are going to form spores. Okay, when the condition becomes unfavorable, again spore. So they come together, they sit calmly. When the condition gets a little favorable, again they will start forming spores, and that is how these people are going to continue. Now I'll tell you here exactly. Under suitable condition, they form an aggregate called as plasmodium. They form an aggregate called as plasmodium. Okay, the mature plasmodium forms young sporangium. Now all of you know what is a young sporangium. Okay, it's a structure like a fruiting body which can hold your spores inside, right? A sporangium. Okay. Now, the mature sporangium bears spores. So, inside this, what are we expecting to see? You're expecting to see spores. So, when the condition gets favorable, this can be scattered everywhere. Okay. Now, the spores undergo meiosis. The mature sporangium outbursts and release the spores. The spores possesses true walls. So, this is one of the important things that you have to remember. The spores are going to have true walls. Why? So that these people can go wherever they want. Okay, if I have a protection, if I have like 2 blackheads, I'll be like, yes, let's go. Okay, so like that, the spores have true walls. They are extremely resistant to survive for many years, even under adverse conditions. Okay, the spores are dispersed by air currents. The spores germinate and differentiate into flagellated and amoeboid spores. So they don't know, these people are similar to protozoans. Why? Because of the movement. Okay, because of the movement. In protozoan, students, that is animal like, what do we talk the most about? Locomotion. Right. So here, flagellated and amoeboid spores. So with the movement, we are connecting them to protozoans. Okay. Now, zygote again develops into mature plasmodium. Any doubts in this? So if you want, we can see the cycle here. Okay, look here, students. How did this formation happen? How did this plasmodium formation happen? It is not your malarial plasmodium. This is a different plasmodium. Okay. So look here. First, let's say a plasmodium, mature plasmodium preparing to young fruit, young the, did I say fruit the young the? Okay, so feeding plasmodium. So this is what happens when the conditions are favorable. Okay, when the conditions are favorable, they aggregate and they form the plasmodium. Now the plasmodium will be maturing, it will be ready to start growing the sporangium. So you can start seeing small sporangium. Inside the sporangium, what do you expect? Spores will be formed. So when the condition becomes favorable, this can outburst and it can be carried away by air currents. Okay, so spores are there. Inside that you can see kuti spores are there. Now they have undergone meiosis, germinating spores are formed, amoeboid cells are there, flagellated cells are there. So two types are formed. The spores are going to form what? Amoeboid and flagellated, and then again fusing will happen, syngamy, formation of zygote, and again the cycle will continue. Now you don't have to worry, ma'am, should I learn this entire cycle? Not at all. But in our batches and all, we teach about this cycle. Why? So that there's a connection. So simple, students, what and all you have to study. During unfavorable condition, they form an aggregate called as plasmodium. Okay, that plasmodium will be waiting. Then the condition will become favorable. During that time, he is going to slowly form the sporangium. He is going to mature and he is going to form the sporangium. Inside the sporangium, who is going to be there? Spores are going to be there. And what will they do? The sporangium is going to burst open and the spores are going to be carried by air currents. And that will go and fall somewhere and they will start germinating. That's it. Okay, that's it. Okay, Hema's head is breaking out of tension. Why Hema? It's not even that difficult to remember. Okay. So first, so I will tell you, I'll give you another example. Okay, I'll give you another example. Okay, let's say we did a house party. Okay, let's say I called all my friends home. Okay, we are going to do a house party. Now what will happen? I'm doing a house party. We are all together. That is, condition is favorable. So we all came together. Okay, so when you all come together, I will call what? Plasmodium. Now students, suddenly 404 restriction happened. That is, people are not supposed to come together. Okay, so suddenly police started checking everywhere. If police start checking everywhere, then what will happen? We are all going to hide, we are all going to sleep, and we are all going to switch off all the lights and not play any music, correct? Now again, when the condition becomes favorable, what are we going to do? Slowly we are going to lift our head. So here what is going to happen? Slowly this is going to mature and form sporangium. Okay. And when we know that 404 is completely removed, what is going to happen? Inside the sporangium, spores were there. These spores will get released by air currents, correct? So we will all start going out. Instead of doing house parties, we will go out and we will go to cafes and sit, right? So it will be released with air currents, and wherever we go and land, germination will happen. That's it. That's it. Okay, easy peasy lemon squeezy it is. Okay. Now protozoans, come on. Let's do it. Yes, yes. Wow, not bad. In half an hour, we have finished. Okay, we are going to finish. Come on. Okay. So, more energy you give me, I'll be able to finish faster and do better. Okay. Come on. Now, students, protozoans. So, what do we know about protozoans? We have to know that protozoans are divided into 4 of them. Okay. Protozoans, again, we call this animal-like. Mainly because of movement. Animal like, mainly because of what? Locomotion. Okay, the bodies that are going to help us in different kinds of movement. Okay, now these people, 4 of them are there. Who are the 4 of them? Amoeboid we have. Okay, amoeboid we have, ciliated. Okay, next whom do we have? Flagellated. Last we have what, students? Sporozoans. Correct? Now, what do you know about each of them? What do you know about each of them? Amoeboid, ma'am, amoeba, right? Amoeba. So, here you go. This is an amoeba. What do this amoeba have? Ma'am, very simple, ma'am. They have pseudopodia. So here under amoeboid protozoan also, I am telling that the movement is going to happen with the help of pseudopodia. Now what you have to remember here, you have to remember the examples. Here students, the example is going to be Entamoeba histolytica, which again you will be discussing in human health and diseases. Okay, histolytica. Okay. So Entamoeba histolytica. Pseudopodia is there. Pseudopodia is there. Now one more thing is marine amoeboid protozoans will have silica. Okay, silica deposition. So that you can remember. Extra point. Marine one will have silica. Okay, silica deposition just like cryophytes. Done? Easy? Yes or no? Yes, they move like amoeba. How does amoeba move? With the help of pseudopodia. So here also it is going to be with pseudopodia. Correct? Am I right or wrong? Yes, ma'am, you're right. Or am I wrong? No, ma'am, you're not wrong. Okay, so yes, this one thing you remember, the marine ones will have silica deposition. Okay, next one, students, we are going to ciliated. Ciliated, the example is going to be paramecium. It is going to look like the sole. Sole is there, no, inside the shoe. Now, I don't have shoe, students. So, inside the shoe, sole will be there. So, I'll draw and show something like this. Like this. It became very underweight. Okay. Like this. Sole of the shoe looks like this. Yes or no? So, these are going to be completely surrounded by cilia. Okay, like this. Hair-like structures. What are they going to do? Ma'am, locomotion. They're going to bring in the food particles. Right? So, same thing is going to happen. Okay, yeah. So, this is your paramecium. Now, students, can you see the hair-like structures? It's kind of small. Yeah. So, this paramecium, students, they have a special structure which is called the gullet. Okay. Gullet. Some people pronounce it differently like this. Like this. One entry. Okay. Like one entry, they have a structure where the particles, all of it will enter through. Okay, where the food and everything will enter through a gullet kind of a structure. Now, one more most important thing about your paramecium is that these people have two nucleus. Okay, one macronucleus and one micronucleus. Okay, they're going to look like that sunflower, but I'm not going to draw that and complicate. So, just remember this much. Okay, what are these people going to have? Cilia. Okay, hair-like structures. And students, what will be the example? Can anyone tell me the example? They cause sleeping sickness. Okay. Trypanosoma. Sleeping sickness. Yes. Suhas here, ma'am. Hi, Suhas. Okay. Yes, cilia helps in bringing food into the gullet. Okay, into the gullet. Very good. Okay, so macro and micronutrients, can you remember? Sorry, macro and micronucleus, can you remember? Should I write it separately, students? No need, no? Okay, students. So sorry. I wrote instead of flagellated this one. Okay, here paramecium. Example, paramecium. Okay, yeah. Okay, flagellated is going to be trypanosoma. Okay, it's a small, like an oral cavity, dear. It's like an oral cavity. Okay. Yes, it's like a gullet is like an oral cavity, a small way for this water, for the food particles to enter. So cilia will be helping them to allow this entry. Okay. Next is going to be flagellated and sporozoan. So students, flagellated, nothing to draw. Okay, flagellated, nothing to draw. Only remember the example. Flagellated means what? Flagella will be present. Correct. So here it is going to be flagella. Flagella will be present for movement. And example here is what you're going to write, trypanosoma, which is sleeping sickness. Okay, sleeping sickness. And the last one, sporozoans. Sporozoans means their main movement is going to happen with the help of what? Ma'am, it is going to happen with the help of pores, right? Here also same thing, students. Here pores are going to be the main people who is going to help, and you have already studied in human health and diseases sporozoids. That means where? Malaria. Malaria is caused by whom? Plasmodium vivax. So here the example is going to be Plasmodium vivax. Can you remember this much? Plasmodium vivax, which causes malaria. Okay, malaria. Done? All of you understood? Yes. Okay, Plasmodium, which causes malaria. So spores are the movement, no? That is how they are going to spread. Now one place we have amoeba, one place we have flagella, one place we have cilia, but this fellow is going to take the help of whom? Spores. Spores are going to spread. Sporozoids. We have learned it in malaria, Plasmodium vivax. So this much only you have to remember, students, in this entire topic. Only this much you have to remember. Can you all remember this much? Yes or no? So I'll show you ciliated structure. So can you see the oral groove, which is called the gullet, through which the food particles are going to enter? You have a macro and a micro nucleus. Okay, two nucleuses are present. Okay. Yes. Nothing there. Sporozoans means spore. Spores are going to be there. That is the movement that they choose. So in Plasmodium vivax, that is your malaria also, sporozoids are there. That sporozoids only enter into the blood of a host, okay, and that is going to spread, go to the liver to RBC, burst open the RBC and release the hemozoin, a toxin, and again it will continue the entire life cycle. So same way here also sporozoans do the same job. Clear? Yes? Okay. Now students, I've just kept the diagrams here. Okay. Now let's do fungi. Ready for fungi? All of you? Okay. Yes. Let's drink some water. Okay. So far, any doubts? Last, I'll make you solve questions. Okay. Last, I'll make you solve questions. Okay. Perfect. Everything is clear? Crystal clear? NEET result today? No way. The court had to give this one, no? I don't think so. Yes, those who haven't marked your attendance, please mark your attendance as well. Okay. Wait, let me check. No results today. And they will not publish it at this late also. Okay, come on. Let's do this. Okay, fungi, students. What do we know about fungi? First of all, this fellow was very difficult to, be placed somewhere. So, because why? These people are saprophytic, parasitic, heterotrophic nutrition, eukaryotic. Again, some exceptions are there, unicellular, multicellular, yeast is there, that we will come into. Okay, so fungi, when we talk about, the first thing that you remember is what? These people are eukaryotic. Okay, eukaryotic. They are multicellular. Again, exception is there. Okay, multicellular. They are heterotrophic. What else do you remember about them, students? This is what we remember about them, right? They are eukaryotic, multicellular, and they are heterotrophic organisms, right? Yes? Very good. Now, Students, here let's break down each word. Eukaryotic means we already know these people are going to have all membrane bound organelles plus the nucleus. Okay, no doubt in this. Here we have to write about an exception. Okay, we have an exception. That is who? Exception is your Saccharomyces, okay, yeast, right? Yeast, which is again, yeast is two types. We have baker's yeast and brewer's yeast. You'll study this in your microbes chapter. Baker's yeast and brewer's yeast. Oh, my tongue. Okay, brewer's yeast. Okay, two types of yeast. Now, when we talk about heterotrophic organism, remember that we are not just labeling them as, okay, they are dependent on other organism, but they have two main way of, doing heterotrophic nutrition. What are they? One is going to be saprophytic. That is the main thing. That is, these people are going to feed on dead and decayed organism. That is why if you leave a bread in your, in your room for like 3-4 days, or you buy the bread and you forgot to eat it, okay, what we do is we buy the bread. Okay, first 3 days we'll be very energetic. Yes, I need to eat bread and omelette. I need to eat bread and jam. After 1-2 days, we'll get bored of bread. We'll forget it. The last 3 pieces, which are the most ignored people in this entire world. Students, they are feeling no one can understand. Okay, those 3 pieces are going to go through what? This fungi is going to do saprophytic. Okay, there's going to be saprophytic nutrition. Why? Because these people are going to feed on dead and decayed. Yes or no? Yes or no? Do you agree? Last, your mom has to take it and throw with you. Then only we will notice that, oh, wait, did I buy bread? Dead and decayed. Next is going to be parasitic. Disease causing. Okay. Parasitic nutrition. So, these are the 2 types of heterotrophic. One is saprophytic, one is parasitic. That is, these people are going to be feeding, or they are going to use a host. Okay, use a host. Now, associations are there. Don't get confused. Associations is where fungi is helping an algae, fungi is helping a higher plant, mycorrhiza, lichen. That is different. Okay, association is completely different. But this is clear. Now, students, look here. If you leave your bread for a few days, you will start seeing a bread mold. Okay, and this is your toadstools, and this is your rich source mushroom. Okay, agaricus. So there is good ones and bad ones. In everything there's good ones and bad ones. Okay, now here they have given 2 examples in your NCRT. One is wheat rust caused by Puccinia and white spots seen on the mustard leaf. Now students, what happened? This was a Petri plate. Okay, this was the Petri plate, and in this Petri plate, this Petri plate was unwashed. Okay, this Petri plate was unwashed, and fungus were growing in this. Okay, there was fungus growing. There is bacteria also. Okay, first bacteria was growing because this was unwashed. Fungus started growing, and what happened? The scientists started noticing that the region where this fungus is growing, bacteria is not at all there. There is a zone. Okay, a zone is created, and in that zone he can notice that the bacteria is not growing around this fungus. That is when he realized that maybe this has some properties. Okay, this has some properties that will kill the bacteria, and that is where your antibiotics, okay, penicillium, came into existence. Okay, clear? Yes or no? Yes, clear? Penicillium, Puccinia, all of it is clear, I hope. Okay, very good. Okay, now students, this fungi are cosmopolitan. Okay, cosmopolitan means they are present everywhere. Now in your room, in your hand, okay, every single place this will be present. They are cosmopolitan. They are present almost every single place. Okay, every single place. Now we actually have to study more about fungi. Other than example, we have to study about their structure very importantly. Now their structure, when you talk about students, they have a thin, long, slender structure which is called the hyphae. Okay, they are made up of thin, long, slender structure which is called the hyphae, and many hyphae, a network of hyphae will form what? Mycelium. So like this, like this, like this. So like this, like a random, okay, like this, a network, a complete network of hyphae, okay, a complete network of hyphae is called what, students? It's called mycelium. It's called mycelium. Okay, now remember this hyphae can be of two types. What can hyphae be of? Two types. Okay, very good. All of you are answering. I can see it in the chat box. This hyphae, students, can be of two types. Remember this. First type is going to be called as aseptate. What is a septa? Septa means like a boundary, like a partition, right? So the first category that you're going to study here is aseptate. So let's say this is your hyphae and you will notice that they do not have any proper partitioning, okay, or any compartmentalization. They're just present like this, okay. On the other hand, you will notice that there are structures like this, okay, where there is proper partitioning. I will show it to you now. Wait. Okay, like this. Correct? Like this, proper partitioning is there. Okay, so I call the one which does not have proper segments or anything as aseptate. Okay, what do I call this as, students? Aseptate, aseptate or cenocytic. Cenocytic hyphae. Okay, this is called as what? Septate. This is called as what? Septate hyphae. Now students, this aseptate or cenocytic is usually seen in lower fungi. It's usually seen in lower fungi. And this one is seen in the higher fungi. Higher fungi. Clear? Like Basidiomycetes, Ascomycetes, we are going to see this. Perfect? Clear to? Yes, there is no cross walls and there is cross walls. Okay, these are the cross walls that I'm talking about. Okay, cross walls. Done? Easy peasy lemon squeezy. Sorry. Okay, I hope it's clear. Yes. Now students, remember these people's cell wall. Okay, these people's cell wall are made up of, I'm writing it here near the structure only. Cell wall is made up of chitin. This you already know. We have already studied in R.H. Whittaker's box. Chitin and polysaccharide. Okay, everything about structure. So we will write it together in one page. Okay, polysaccharide. Done. Something is wrong with the spelling, no? Polysaccharides. Okay, yes. Sometimes when we write on the board, no, we feel it's correct, but it might not be correct. Done. Pakka? All of you understood? Yes, Phycomycetes are aseptate. Yes, we will come into that. Okay, we will come into that entire topic. Okay, this thermocol reflection is too much, no? Here, any white thing will reflect, students. Now it's better, right? Okay, 1 second. Let's try. Okay. Yes, the dots are nucleus. Okay, the dots are nucleus. Okay, now it's not visible, no? We have to do jugad. Okay, jugad work we have to do. Yes, done. Okay, so this is about the structure. Now talking about the reproduction. So students, remember that your fungus, okay, your fungi is going to reproduce by 3 methods. One is going to be vegetative method, one is going to be asexual reproduction, one is going to be sexual reproduction. So in vegetative method, we will study about the normal things, fragmentation, binary fission, okay, budding. But when we talk about asexual and sexual reproduction, understand, there we talk about different types of spores. So I do not want any of you to get confused. Listen to me carefully. And also there is a cycle of events that happen during the sexual reproduction. So these are the two things that most of the students get confused, but let me tell you, it's going to be very easy to understand. Okay, so students, what is going to happen? Your reproduction in fungi, as I told you, it's going to be in 3 types. First is going to be vegetative First is going to be vegetative reproduction. So whoever knows the answer, please type that in the chat box, okay? Vegetative reproduction, okay? Next one is going to be asexual reproduction. Asexual reproduction. Okay, and the third one is going to be sexual reproduction. Sexual reproduction. Now, vegetative reproduction means, students, what and all can you think about vegetative reproduction? Come on, tell me. Come on, tell me. Fragmentation, fission, budding, very good. Okay, so here when we talk about vegetative reproduction, we think about budding, and here we mainly talk about your yeast. Okay, budding, fission, then what, students? Fragmentation. Okay, fragmentation. These are the three things that you will be studying under vegetative reproduction. When you think about asexual reproduction, please remember you're talking about spores. You're not talking about any other method, you're talking about spores. So here you're going to come across conidia. Okay, conidiospores. So I'm just writing conidia. You're going to come across zoospores. Okay, and lastly you're going to come across sporangiospores. Sporangiospores. You don't have to go much detail about these spores. Okay, just know this. Okay, where conidia is there, where zoospores are there, where sporangiospores are there. Okay, now when you talk about sexual spores, students, you start with oospores. Then you come to ascospores, which are specific to ascomycetes. Then you talk about basidiospores, which are, related to basidiomycetes. Okay, basidiospores. Can you remember this much? Isn't it easy? So vegetative reproduction, we talk about budding, fragmentation and fission. When it comes to asexual spores, we speak about zoospores, conidiospores, sporangiospores. And when it comes to sexual reproduction, we talk about oospores, ascospores and basidiospores. So ascospores and basidiospores, you do not even have to think a lot, because it is about ascomycetes and basidiomycetes. Okay. Now when you come about conidia and budding, fission, fragmentation, we will go in detail. Okay. Clear to all of you? So look here, vegetative reproduction, asexual reproduction is by spores called conidia or sporangiospores or zoospores. Look at conidia, students. Here remember, conidiospores are exogenous. What is exogenous meaning? It is outside, right? It is going outside. So let's say the structure is like this. Okay, the structure is like this. You will see conidiospores like this. Understood? Like outside, exogenous. On the other side, you can see sporangiospores, where there is a sporangium. Inside the sporangium, the spores are going to be present. Zoospores, if you notice, you can see that these people are like motile and they just move around. Okay, they just move around. They are motile structures. Clear? Yes? Okay, now look here. See, remember, do not get confused. For everything, you will have sexual reproduction, asexual reproduction, vegetative reproduction. Okay, so all the three are going to be present in different classes of fungi. So Phycomycetes itself, you will study one sexual, one asexual, one vegetative. Like that, each one you are going to study like that. Okay, got it? Done? This is clear to all of you? Okay, now here, look here, students, you will notice that ascospores, when I talk about, there is a structure which is a sac-like structure called as ascus. Inside that, there will be ascospores. So if I have to draw an enlarged structure and show you, no, I will just show like this. See, look here. This is not how I wanted it to be. Like this, yeah. Okay. Okay, now look here, students. Let's say we will just put them in a box. Okay, we put them in a box. Because many students get confused with this one thing. But to be frank, it's not that confusing. So students, when I talk about ascospores, remember, there's a sac-like structure which is called the ascus. And inside that, there is going to be your ascospores. And that's why this is also called as the sac fungi. Okay, sac fungi. So this one, you tell me, can you understand? So this is the ascus. Okay, inside that there is ascospores. But on the other side, you have basidium. Okay, basidium, where your basidiospores are going to be present on the top of the structure. Like this. Okay, 4 of them are going to be seen. So it is going to be like this. Understood the difference between basidiospores and ascospores? I hope it is clear. Okay. Thank you so much, Padmavati. Ma'am, both oospore, zygospore. Yeah, we will come into that. Okay. Hercules. Don't get confused. We will come into that. Okay. So what did I say you? This is ascus. That is a sac-like structure inside which the ascospores are going to be present. And this is your ascospores. Any confusion in this? Any confusion? Confusion. That song is coming, students. But I don't know the lyrics. Basidiospores. Okay, basidiospores. Clear? And this is going to be the basidium. Okay, this is going to be the basidium. Why I am drawing diagrams? So it will be more clear for all of you. Okay, I can rush with the chapter, but what is the use? What is the use of it? Okay, this is the difference. I hope this is clear to all of you. Now, yes. Sexual reproduction. I told you there are some events that take place. Okay, there are some events at which the sexual reproduction is going to happen. See, now I am just going to introduce to the spores. I'm just going to introduce you to the events, and then by one, depending on each fungi, Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes, we are going to learn individually. Okay, yes. Now students, sexual reproduction, remember, it is going to happen in a events of cycle of events or events of cycle. Oh, English is not Englishing. Cycle of events, I guess. Students, tell me. All the encyclopedias in the class, please open up. Cycles of events, okay? Yes, this is what is correct. Sounds correct. Okay, cycle of, tell me it is correct. Cycle of events, okay? Content is correct, but then I don't know the heading of it. First one is going to be called as plasmogamy. What is it going to be called as? Plasmogamy. Again, I'm going to explain it with the help of a diagram, so don't worry. Okay, let's take two different colors. Okay, this one. Our teachers also used to draw like this and teach us, students, once upon a time. Okay, so this is how we have two structures, okay, like this, right? This is the initial stage, okay, where two of them are going to come together, right? Now, what is going to happen? First, what is going to happen? Ma'am, first, what is going to happen? Protoplasm is going to mix. So, you go like this. Okay. So, it's not an easy task, students, to draw. Okay, like this. Protoplasm mixing is going to happen. Okay, so the nucleus is going to remain intact here. Okay, here also same thing. Protoplasm is going to movement. Okay, movement of the protoplasm. See, I cannot draw 3D. I'm trying my level best to draw and make you understand this. Okay, clear? I hope at least you can understand what is going to happen. Okay, so students, in plasmogamy, what is going to happen? It is going to be fusion. Okay, fusion of protoplasm. Fusion of protoplasm between two motile or non-motile, okay, or non-motile gametes. Correct structures? Yes or no? Correct, ma'am? Correct, ma'am. Okay, very nice. Okay, two motile and non-motile gametes. That is what we see in your first stage. Okay, so this is your protoplasm, where the protoplasm fusion is going to happen. Okay, protoplasm fusion is going to happen. After protoplasm fuse, what would happen, students? Ma'am, after protoplasm fuse, then it should be definitely what? The nucleus should come together. What do you call that, students? Karyogamy. Karyogamy. That means the nucleus has to come together. So let's take it to the next stage. Okay, let's take it to the next stage where it is karyogamy. Okay, karyogamy. Next stage, that is here protoplasm mixing has happened. Now these people are going to come into one entire cycle, right? So here I'm just drawing it in two colors, so you understand it properly. It's not necessary to draw in two colors. We can just draw it in one single color also. Geometry is not geometry. Yes. Yes. Okay. So here, students, see, two situations will come. Okay. Two situations. Yes, gametes, sexual reproduction, no dear. So gametes are going to be formed. Correct or not? Yes. So students, remember, two situations are there. Okay. This situation here, where you can see two nucleus, right? This is nucleus 1, this is nucleus 2. We call this condition as dikaryon stage. What do you call this condition as? Dikaryon stage. Why do we call it dikaryon stage? Because here we notice that the protoplasm fusion has happened, but the nucleus have not got fused. Right? We will notice that there are two nucleus in this stage. This is why we call it the dikaryon stage. Okay. Now, if you notice here, students, a single nucleus is going to be present. Okay, single nucleus is going to be present, and this is where karyogamy has happened. Okay, karyogamy has happened. So I have done it like in different colors, but you can just draw it in one color to understand. Karyogamy, that means fusion of the nucleus has happened. Okay, fusion of the nucleus has happened. So is dikaryon stage clear for all of you? Because later you're going to study about it. That's why. Protoplasm fusion is happening in the first scenario. Okay, this is the first scenario. This is the second scenario where the karyogamy fusion is going to happen. Yes, very good. 2n. Okay, 2n. Okay, even without the line, we can just write it. 2n. Okay, so here karyogamy means what? Fusion of nucleus. Okay, fusion of nucleus. Yes. Done, done. Is this clear? Yes, I'll move to the side for you to all understand. Yes, this is clear, students. Yes, dikaryon stage also is clear, I hope. Yes, students, gamete formation. Normally, you know, no sexual reproduction, gamete formation has to happen. So that will happen. Okay, that will happen. The last one is obviously zygote. Okay, zygote formation by meiosis. So that we don't have to write specifically, but here I'm just writing zygote formation by meiosis. So remember, in your asexual spores, no, we talk about mitosis. Remember that also one thing here. When we talk about your asexual reproduction, no, we mainly here focus on mitosis. Okay. So that is why here we say asexual spores. Here what will we talk about? Meiosis, right? Here mainly we will talk about meiosis. So this is the proper sexual reproduction. Got it? Yes. Now, I hope this is clear. So students, 3 cycles are there. What and all? Plasmogamy, karyogamy and zygote formation by meiosis. These are the 3 things that will happen. Now, types of gametes. That is, when sexual reproduction is happening, we know that 2 types of gametes will be there. One from the male, one from the female. But here in fungi, there is more types of categories we have to study. First one, students, depending on their shape, size, we have 3 categories. First one is called as isogamous. Okay, isogamous. Second one is called as anisogamous. Anisogamous. Third one is called as oogamous. Oogamous. Okay, just with the structure we are going to learn it. We are not going to break our head into learning more about it. See, isogamous means both of them will be of the same shape and both of them will be motile. Okay, like this. One scenario, another scenario where both are non-motile. This scenario will be less because again, they have to travel. So this scenario where their size, okay, size will be same. Okay, size and shape, okay, will be same. Okay, and both will be motile or both will be non-motile. Isogamous. Iso means same, right? Iso means same. Yes, Harshita, I'll be finishing the chapter today. Okay, so that is done. Clear to all of you? Now, anisogamous. Anisogamous means what? Ma'am, one will be bigger maybe. Okay, one will be bigger. Okay, understood. Another one will be smaller. But both will be motile, students. Okay, both will be motile. So here what is going to happen? Size and shape will be different. Okay, size and shape will be different, but their motility will be the same. Okay. This is anisogamous. So opposite of this. Just write opposite of that. Okay. Now, one second, students. Okay. Now here, okay, coming to oogamous. Oogamous, we are aware. Okay, we are aware of it. Why we are aware of it, ma'am? Because this is the same thing that is present in humans as well, where you have the female, which is big and non-movable. It does not move. And then we have the male, which is small and movable, right? So here we notice that the size also is different. Motility also is different. Clear? So here we notice that the female is going to be, like, you know, female is going to be non-motile. Female is going to be non-motile. Okay, and the male is going to be what, students? It is going to be motile and small. So it can travel easily. Done, done, done. Easy, no? So easy, right? So these are the different types of gametes. Any doubt in this, students? Any doubt in this? Any doubts? No. Oogamous, yes, very good. Clear, ma'am. Done, done, done. Dikaryo. Yes, very good. Perfect. Perfect. I hope all of you understood this. Okay, we will now learn the different classes. Okay, different classes. Hi, Nishant. Okay. Yes, will you be finishing the chapter? Yes, I will be finishing the chapter. Okay. Done. Okay. Chalo, let's go forward. Okay. Now, students, kingdom fungi is divided into what is this? Oh, color. Which color do you choose? 1 second, students. Okay, yes. So, kingdom fungi is going to divide into Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes. Now, ma'am, how did you do this classification? I did not do. Okay, whoever did the classification did it based on the morphology. Morphology. Question has come, okay, in the previous year from this. Fruiting bodies. Fruiting bodies and spore formation. Okay, so based on these three, they have done the classification and they have classified it into Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. Clear to all of you? Yes or no? Till now, no doubt. Very nice. Okay, now we will go through NCERT line, students. Okay, for each of the classes, we will go through NCERT line. Okay, so members of Phycomycetes are found in aquatic habitats. Only whatever I am marking, you will study. Okay, and on decaying woods in moist and damp places. So all of you know that fungi itself, whenever we talk about fungi, we should remember that these people are going to be present in moist and humid places. That is why students, when we work out, right, when we go to gym and when we work out, we notice that people get ringworm allergy. Why do they get ringworm allergy? Because of sweat. Okay, because of sweat and the tight clothes, people do not wash after that, or the instruments will not be sanitized. So because of that, the person will have fungi, like fungus allergy, and they say no, use that candid powder and all of that. So that is because of this main reason, because they will grow in moist. Okay, now on decaying woods in moist and damp places, or as obligate parasites on plants. Okay, so that means these people are disease causing as well. Okay, now the mycelium. So we have started talking about the mycelium. So I'm going to highlight all of that for you. The mycelium is aseptate. We already discussed that mycelium is going to be, or your hyphae is going to be aseptate in lower fungi. Lower fungi means you only have to remember Phycomycetes. Okay, so they are going to be aseptate and cenocytic. Okay, asexual reproduction takes place by zoospores, which are motile. You already saw the structure now, or by aplanospores. So students, two words they use, just remember this, nothing to be worried about. Plano means, planospores means we use it for motile. Okay, the word motile, instead of motile we use planospores, and we use aplanospores for non-motile. Okay, spores for non-motile. Done? Yes? Okay, now moving forward. These spores are endogenously produced. That means what? These spores, that aplanospores or your zoospores that I am talking about, they are formed inside a sporangium. So, they are endogenously present. Correct? They are endogenously present. They are going to be present inside this. Okay, endo. Endo means inside. Got it? Done, done, done. So, they are present endogenously produced inside the sporangium. Now, moving forward, a zygospore is formed by fusion of two gametes. Here we are talking about your sexual reproduction. So, here the gametes are morphologically similar, anisogamous, isogamous. So, students, here, what is that? Instead of oospores, no, we can also use zygospores. So, here a zygospore is formed by fusion of two gametes. It's formed by fusion of two gametes. Okay, these gametes are similar in morphology or dissimilar. That means they can be isogamous or they can be anisogamous or oogamous. Okay, some common examples are mucor, rhizopus, the bread mold mentioned earlier, and albigo, the parasitic fungi on mustard. Okay, which causes the white rust. So here, students, you people please remember RAM. Okay, this trick I have made before also. RAM. Okay, RAM. So your Phycomycetes example is going to be RAM. R for rhizopus, A for albigo, okay, M for mucor. Done? RAM, please write it in your NCERT now itself. Okay, now itself, and all of you drink some water as well. Okay, please write. Yes, write it now only in your NCERT, now itself. Okay, so remember, students, here, see, one thing that might be confusing for all of you is the word zygospore. See, zygospore, students, it's actually, even though we are telling it is made up of two gametes, we do not say it is made up of female and male gamete. It is like, yes, it is made up of two different things, but it is not like proper, like how we tell about oospores. No, it's not proper like that. That is why we use the word zygospores. Okay, again, please understand there's more to it, like classes depending on the phylums, lot of classifications are there, but just remember, these people are like they're not like the proper male and female. It is not proper male and female gametes type. It is most isogamous types. Okay, like same gametes fusing together. Okay, that is why this word they have mentioned, but they have not explained about that anywhere else. So that is why it becomes confusing. Okay, now RAM. Okay, one second. Okay, wrote, very good. Now, here, next one. Okay, here we have to talk about ascomycetes. Ascomycetes, let's start. Mycelium is branched and septate. Okay, so here we are talking about mycelium which is branched and septate, like this, which we already drew and understood. Now, students, septate, you should remember these are the nucleus structures which I am drawing, and they have proper partitioning. Okay, they have proper partitioning. Now, asexual spores, students, are conidia. Okay, conidia, which are produced exogenously on the special mycelium called as conidiophores. So you would have noticed in that first diagram which I showed you, there were structures like this. Notice, like the small branches types, like this structures were there. On this structure, conidia will be growing. Okay, like this, like a small structure exogenously they will be growing. So I will just label that so you will not have a confusion. Okay, this is going to be your conidia. Okay, conidia, and this is your conidiophores. Okay, conidiophore. Done. Now, conidia on germination produce mycelium. Sexual spores are called as ascospores. This I already explained to all of you. Ascospores, which are produced endogenously inside what? A sac-like structure called asci. Singular is going to be ascus. This also I explained to you some time back, that the structure is going to be like this. Okay, structure is going to be something like this, like a sac-like structure. Inside the sac-like structure, you're going to see ascospores. Okay, this sac-like structure is called ascus. Okay, ascus. Done. This much is clear. Now, these asci are arranged in different types of fruiting bodies called as ascocarp. Okay, ascocarp. Some examples are, students, Aspergillus, Caliceps and Neurospora. Here you are going to remember can. Okay, can. C-A-N. So, Ram can. So far we have studied there. Okay, Ram can. So, you want me to write it? I'll write it quickly. Aspergillus, all of you remember? Aspergillus, again, in the microbes chapter, you will be studying in detail about all of it. Okay, and Neurospora. Neurospora is actually used in many genetic and biotechnology techniques. Okay, so look here, Neurospora is used extensively in biochemical and genetic works. Okay, many members like murals and truffles are edible. Murals and truffles are important for you students. Why? Because in Korea and all, when we make that ramyeon, okay, ramen, ramyeon, okay, they grate it and give it. Okay, it's highly rich, okay, in vitamins, so they give it, but it is expensive. Okay, so that is why murals, it's not morals, it's murals. Okay, murals and truffles, they are edible. They are edible. Okay, they can ask you a question which is edible among these. Now, next one is about your basidiomycetes. The mycelium is branched and septate. Here also same thing. I told you ascomycetes and basidiomycetes, you write the same thing. The asexual spores are generally not found. So this is very, very important. So students, so far which and all did we study? Okay, so far what and all we studied, we will try to write it in one page. Okay, we will try to write it in one page, whatever we studied. Let's see if time constraint happens. I think time will be enough, students. Again, I have 30 minutes now, I'll be able to do it. Yeah. But vegetative reproduction is by fragmentation. Okay, by fragmentation. Now, the sex organs are absent. They're telling the sex organs are absent, but plasmogamy, all of you know, protoplasm fusion is happening, is brought about the fusion of two vegetative or somatic cells. So students, normally what happened? Your gametic, right? Your somatic cells are there, your genetic, we talk about your gametes are there. Now when gametes fuse, we tell, okay, sexual reproduction has happened. Imagine the vegetative cells. Two vegetative cells will do plasmogamy and they will form, spores out of it. So here that is going to happen. Here asexual spores are also absent, sex organs are also absent, but reproduction is happening how? When the two vegetative cells are coming together or two somatic cells are coming together. So look here, of different strains or genotypes, the resultant structure is dikaryotic. Okay, which ultimately give rise to basidium. Okay, dikaryotic means your dikaryon phase. Okay, two nucleus will be there, no? So a stage like this. This, okay, which we just studied now. For those students who like this, but remember, these are somatic cells. Okay, these are not your male and female gamete. These are somatic cells. That is, these people are vegetative cells. Okay, somatic or vegetative cells. Okay, somatic or vegetative cells. Clear? They cause the dikaryon phase. Now, karyogamy and meiosis takes place in the basidium, producing 4 basidiospores. So students, after the dikaryon stage, okay, after the dikaryon stage, just remember, we got a basidium. We got what? One basidium. Okay. In the basidium, what happened after that? We understood that karyogamy and meiosis. Okay. After this, in the basidium, karyogamy, that means fusion of nucleus happened. Okay. And to do the zygote formation, meiosis also happened. And that led to the formation of your basidiospores. But like this, okay, basidiospores which happen exogenously like this. Okay, like this structures we form, that too 4 basidiospores. Okay, the basidiospores are exogenously produced on the basidium. The basidia is the same. Okay, the basidia are arranged in fruiting bodies called basidiocarp. So remember, all of this will be inside a fruiting body. It cannot be exposed like that. Everything will have a fruiting body. Ascocarp will be there, basidiocarp will be there. They are the fruiting bodies. Okay, some common members are agaricus, ustiligo, and puccinia. Okay, smut, rust, and this one. I think students, PAU, P-A-U, this is what we thought, or else opt. Okay, opt. And that T is not there. Okay, opt. So this is the new spelling of opt. Okay, so this anyone you can remember. Okay, puccinia, ustiligo. Okay, ustiligo, agaricus, your mushroom. Okay, and puccinia, rust. Clear? Can you remember? Okay. Clear? Done. So PAW or opt, you can remember one of the words. Now, don't look at this paragraph and feel like, my God, what is this, ma'am? We are going to talk about your Deuteromycetes, which is an imperfect fungi. Everything is confusing about this fungi because nothing is known about the fungi. Okay, that is what has happened. So commonly known as imperfect fungi because only the asexual, okay, only the asexual or the vegetative phase is known. Okay, vegetative phase of the fungi are known. So we do not have any information about their sexual phase. When they got to know that they have a sexual phase, it was confused that this might have been, should have been placed under Basidiomycetes or Ascomycetes. So many confusions happened. So people thought, okay, let's leave it. Okay, just keep him imperfect. So now students, when the sexual form of these fungi were discovered, they were moved into classes that they rightly belong to. Clear? It's also possible that their asexual and vegetative stage have been given one name and placed under Deuteromycetes. Understood? Sexual and the vegetative phase, like combination, they're like, okay, you go under Deuteromycetes. Now the fungi were correctly identified and moved out of Deuteromycetes. Once perfect stages of the members of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. So that is why this fellow became imperfect, because all his perfect stages, once they discovered, they started changing it. Like, when we go to college also, if you're getting 95, all the 95% students will be in A group. That is, class A. Below 95 will be in B group. After that, C, D. So that means what? All the perfect students have been taken into one session. So like that only this also has happened. Okay, pavam fellow guys. All of you please put crying emoji. 2 minutes silence for Deuteromycetes. The Deuteromycetes reproduce only by asexual spores known as conidia. So students, your only person you have to remember is conidia. Okay, same thing. Exogenously formed conidiospores are going to be there. Okay, conidia is going to be there. Clear? Now, here see, conidia, asexual spores. Okay. Now, the mycelium is septate and branched. What is it? The mycelium is septate and branched. Some members are saprophytic or parasitic, while a large number of them are decomposers of litter and help in mineral recycling. Okay. This is important, students, that these people are maximum decomposers and help in mineral recycling. All this can be asked as assertion, reason or statement based question, which we think that, oh, it's such a simple line. Who will give it? NTA will give it. Okay. Now, examples are Alternaria, Coleotrichum and Trichoderma. Trichoderma polysporum. Okay, remember. So, this is cat. Okay, cat. So, students, Ram, can, opt, cat. Opt spelling we have changed. Gopika Ma'am's dictionary production. So, Ram, can, opt, cat. Ram, Phycomycetes, can, Ascomycetes, opt, Basidiomycetes, cat, Deuteromycetes. Okay, can you all remember this much? Yes or no? Okay, students, should I finish virus, viroids and then come back? Okay, I'll finish this and then I will come back to the classes. Okay, yes. Now, virus, students, are most conversed topic, that is it living, is it non-living? Now understand this, virus is non-living when there is no host, virus is living when there is a host. Why? The moment that it comes across a host, it will release its genetic material and it will continue its transcription, translation, everything it is going to do, right? Because they have the genetic material and the protein code. Okay, so now here, remember, virus did not find a place in classification since they are not considered truly living. If we understand living as those organisms that have a cell structure, the virus are non-cellular organisms that are characterized by having a inert okay, crystalline structure outside the living cell. Okay. Now, virus means venom. Virus means what? Venom. Okay. So, virus means venom or poisonous fluid. Okay, contagium fluidum vivarium. So, that we will come into. Now, Ivanovsky in 1892, you have to remember these dates till you have to remember. Now, don't break your head. Ivanovsky recognized certain microbes as casual organisms of mosaic disease of tobacco. Okay, there was mosaic disease in tobacco. Okay. Now, these were found to be smaller than bacteria because they used a bacterial mesh. Now, let's take example. I'm making tea. I use tea leaves. So, when I'm using tea leaves, it will not go through the sieve very easily because my sieve is kutty. It will only the tea powder will go. Okay. Tea leaf won't go. So, here also they used a bacterial sieve and they understood that this fellow is smaller than bacteria, but this fellow has the capabilities to cause disease. Okay. So, through bacterial proof fillers. Now, Bejernik in 1898, okay, demonstrated that the extract of the infected plants of tobacco. So, he extracted the tobacco plant to notice that what is the infection, okay, can cause infection in a healthy plant. So, it is that problematic that if I take an infected, like let's say pus, if I take pus and I spread it on another hand, from here also allergy starts coming, then I know it's very problematic, right? It's spreading faster. So, here and named it a new pathogen. He named it a new pathogen called virus and called the fluid no, that he has extracted to infect a healthy plant. He called it contagium vivium fluidum, which is an infectious living fluid. Understood, students? So far good. So what happened? First, Ivanovsky tested on this mosaic disease in tobacco. What did he do? He used a bacterial proof filters to understand what is this a bacteria. He noticed that they are smaller than bacteria, but they are capable of causing the disease. Bejernik in 1898, what did he do? He collected the fluid from this tobacco mosaic plant, that is the one that has disease, and he tried to infect it on a healthy plant, and he noticed that healthy what an all language. Healthy plant is also getting infected, and he called that pathogen as virus. So, Bejernik is the person who gave the virus name, and he, the fluid, contagium vivium fluidum, and he was like, this fluid is also infectious living fluid. So, we will call it contagium vivium fluidum. Clear? Okay. So, here they have named the new pathogen virus. So, according to NCERT, Bejernik is the one who has called it virus. Okay. Now, Stanley in 1935, okay, showed that virus could be crystallized. Okay, virus could be crystallized, and crystals consist of, that means Stanley studied about the structure of virus. One person found the pathogen, one person told that there is a new pathogen, and one person came down to understand the structure of the pathogen. Okay, crystals consist of large proteins. They are inert outside their specific host cell. Virus are obligate parasite. Okay, chance comes, they will attack. Okay, chance comes, they will attack. Clear? Now, in addition to protein, virus also contains genetic material that can be infectious. Okay, this entire paragraph I will just teach you with the help of a diagram. Okay. Okay. Do I have a slide, students? I do not have a slide. Okay, I will just duplicate this. Okay, today I'll share the notes on both the Telegram channels, Gopika Biology Official as well as Vedantu NEET English Telegram channel. So do not fail to join both the Telegram channels. Okay, all my notes are already there. Okay, this new notes or everything will be updated there. So students, that entire paragraph I'll just explain it with the help of a diagram. What are they telling? They are telling this protein. Okay, this virus has a protein coat. Okay, which is inactive without a host. So that protein coat. Now I am drawing a bacteriophage. Okay, I am drawing a bacteriophage like this. Okay, here we have like a sheath, okay, like a, okay, and then we have a tail fiber, okay, like this, okay, like this. This is your bacteriophage, correct? Now, inside the bacteriophage, what is there? It can be an RNA or a DNA, right? One genetic material is going to be there, okay? It can be a double-stranded structure or it can be a single-stranded structure. So, what is this outer coat? This outer coat is made up of protein, okay? This outer coat is made up of protein, and they are telling that without a host, even this fellow is power. without our mother, how we'll be like very violent. With mother is there, we'll be very silent. Okay, like that only. So here students, it can be the genetic material. This is the problematic one. Okay, and this genetic material can be anything. Okay, it can be DNA or it can be RNA. Both are not present, but again, recently they found a virus where both are present. Okay, leucovirus or something, if I'm not wrong. Okay, now here students, this is the sheath and this is the person. Here this fellow only will attach. Okay, tail fiber. So I'm just writing tail. Okay, this entire thing I will call it the head. Okay, this entire structure, same color, the head. So it has a head and the head has a protein code. Inside the protein code there's a genetic material. Sheath is there, tail is there. Okay, now students, if I do an enlarged structure of this protein, okay, it will have like this structures, which are capsomeres. Okay, like this. Okay, like this, capsomeres will be there. Now remember students, these individual units are called capsomeres. Many capsomeres together forms the capsid. Okay, many capsomeres like this together forms a structure called the capsid, and this is how your protein coat is going to be formed. Any doubt in this? Any doubt in this? So this bacteriophage is who? Who is the bacteriophage? Bacteriophage. Students, it's a virus that infects bacteria. Virus that infects bacteria. Okay, I want all of you to comment and let me know how is the class. Okay, all of you come and go. Okay, that shouldn't be the attitude. You should also be, you should talk to me in the comments. Otherwise, I will not remember you people for, like, I have a very good connection with my last year students and last year students. Okay, I still text them. We still text on Instagram. That is, whenever they have a problem, they always text me, and whenever they come across any of my class, they take a minute to comment. So that is the kind of connection that should be there in the online medium. Okay, so I know many of you do not like that. Many of you are like, ma'am, I'm scared to text, but I want all of you to comment because I want to know your names. I do not know many of your names in this batch. Okay, only if you talk, connect, I will know your names. Now few names I know. Okay, their names it's stuck in my head. I will never forget it. Okay, so we should build that connection. One teacher student beautiful relationship should be there throughout this period. Otherwise you're going to get bored and you're going to be very lonely in this journey, because I'm going to talk to the camera. I do not have any connection with you. I will just come, teach, go. But if I have a connection with you, every comment of yours, every message of yours, I will be responding. So we will have a healthy. So whenever you have a problem, you can always tell me, ma'am, please message me, ma'am, I'm going through this. I will always be there. Okay, Ria, clear? So students, bacteriophage is the virus that infects bacteria. This is how the structure is going to look. Okay, now we have only 3 topics to finish. Okay, we will be finishing it now. Students, viroids. In 1971, okay, 1898 you remember. Okay, now we are talking about 1971. Diner, okay, discovered a new infectious agent that was smaller than virus. Okay, it was smaller than virus, caused potato spindle tuber disease. That is, students, normally how does the potato looks like? Ma'am, it will be round, goal, right? Goal potato will be there. But students, because of this viroids, okay, because of this viroids, the potatoes started becoming spindle shaped, like how your cucumber is there. Now, obviously, a farmer who is selling the potatoes will not start earning any money, because no one is going to buy that potatoes, right? So, potato spindle tuber disease, it was found to be a free RNA. That means they noticed that it was just a genetic material. It did not even have a protein code. It did not even have anything. Okay, yes, okay, very nice, Hema. It lacked the protein code that is found in virus, and they called it viroid. So, it was what? Naked genetic material. It was what? Naked genetic material. Clear? The RNA of the viroid was low molecular weight, okay, and then came prions. Okay, in modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of an abnormally folded protein. Not a normal protein, but an abnormally folded protein. The agent was similar in size of a virus, and these agents were called as prions. Okay, now they cause the most severe diseases in human students. They would directly attack our brain. That's why it's called as encephalopathy. Cephalization. Whenever the cephal word comes, cephalochordata, we connect it to the head. Okay, the most notable disease caused by the prions are bovine spongiform encephalopathy. Encephalopathy. See, encephalopathy, brain. Okay, commonly called as mad cow disease. So this was seen in cattle, students. They will have continuous burning sensation. They will be, continuously trying to, run, move. It's very like, you'll notice that slowly they will start getting paralyzed. Their both the, legs will start getting paralyzed. So that kind of a disease. Okay. Now that can happen in humans also by, let's say, consuming the meat also we can have it. Otherwise, the mad cow disease, and in humans, its analogous variant, okay, is going to be Creutzfeldt-Jakob disease, okay. Jacob disease, some pronunciation is there, K pronunciation is there. So you can write CJD. This is your new NCRT. 2025 NCRT full form is given. Before only CJD raised to write. Now full form is given, okay. Creutzfeldt-Jakob disease in humans. Clear? Yes, I hope this is clear to all of you. Okay, let me... Done, students? Perfectly clear? Now we have only lichens, okay? Lichens. See? So, first was viroids. So, viroids, what happened, students? Only the genetic material was there. Free RNA was there. Okay? Free RNA. And this free RNA caused what? Potato spindle. Tuber disease. Okay, so normally the potato would be round, right? Goal mole. The potato used to be like this, round potatoes, and then slowly it started becoming spindle shape like this. Okay, spindle shape. So this was the first one. Next one is about your prions. Okay, this one, students, what has happened? Remember here, dates and all we will study at the end of the year. Okay, now we cannot study. Yeah, so here, students, what happened? Normally your protein would be like this. Okay, protein would be like this. Suddenly they started noticing that there was an abnormal folding in the protein. Okay, okay, there was an abnormal folding in the protein. That protein, I mean this one, okay, went and attached to where? To the brain. Okay, to the brain, causing bovine spongiform encephalopathy. Okay, and second thing, what did it do? It went and caused mad cow disease. Okay, mad cow disease in cattle, and the analogous variant is CR-Jacob disease in humans. Clear? Okay, this is what happened. Okay, this was just protein. Okay, this was just protein. This is normal protein, and they were all parasitic. Okay, so only free RNA, only protein, and that led to the formation. Okay, this was just the RNA. Okay, just the RNA. Done, students? I hope this is clear. Now, last one is lichens. Nothing much about lichens. Two words you have to remember: phycobiont and mycobiont. Okay. Phycobiont and mycobiont. Okay. Phycobiont is the algal part. So, all of you know, lichen is a symbiotic associationship between fungi and algae. Okay, and mycobiont is going to be the fungal part. Mycology, so fungal part. So students, very simple, students. Fungus used to give the shelter, protection, we can tell. Okay, and algae used to provide the food. This was the symbiotic relationship, okay? And remember that lichens, okay? This combination of phycobion, mycobion, whatever together, no, lichens. These lichens are only present in a pollution-free place, okay? Pollution-free. That means these lichens are good indicators. Okay, they are good indicators of pollution. That's why you will notice that there are trees in your college inside. Okay, like in your middle campus is there, no students? Okay, mid campus. Yes, hi Meghana. So middle campus students, there will be, you will notice that trees are there where lichens are present. But you will not notice the same tree if it is outside. No, you will not notice lichens. Why? Because they are good indicators of pollution. Where pollution is there, the lichens will not be present. Clear? These are associations of your fungus. One more association is your mycorrhiza. Okay, mycorrhiza. Students, mycorrhiza, a genus called as glomus. Okay, it's just write fungi. You can just otherwise just write fungi plus higher plant also. Fungi plus higher plants. Okay, so this is also one kind of an association. Okay, one kind of an association. Clear? So I've completed the chapter. Again, same thing. One provides the nutrients, one provides the shelter. Clear to all of you? So this is your phycobion and mycobion. Students, remember the lichens, when you notice, no, they are going to be something like this. Okay, like this big patch it is going to be. And if you notice very closely, in the center, you will start seeing the green color dots. That is where your algae will be there. Now, again, lichens are of different types. Okay, different types, depending on the algae, the lichens are going to be different. Okay, depending on the phycobion, your lichens are going to be completely different. Clear to all of you? So I've given a summary here. Okay, I've given a summary here so that all of you can just, all of you can just read through it. Okay, this is like the overview of the entire chapter. Okay, done. Okay, let's do some few MCQs very quickly. Okay, after that Shreya sir will start. I'll just tell sir to start at 10 minutes late. So you get a little break. Okay, 1 minute students. I'll just tell sir. Okay, come on. Yes, answer this quickly. First question in 5-kingdom classification, which single kingdom contains, can you see this? Blue-green algae, nitrogen-fixing bacteria and methanogenic archaebacteria. Students, quickly answer. Okay, I think I will be able to see the chats a little late. Students, when we do revision, no, we will revise that Deuteromycetes. So just remember the trick. Okay, remember the trick. Ram can opt, this is the new spelling, okay, we only made opt. Okay, T is silent, so just don't write it only. Cat, that means he can opt for a cat, like dog and cat is there, he will go for a cat. Okay, so this you please remember and keep, right? Yes, very good students. Monera is the answer. Monera is the one that has all of this. Okay, fungi usually stores the reserve food material in the form of, okay, this is a information that I wanted to give you, so that is why I put the question for it. I always do that. Okay, this is an additional information or an information mentioned in your NCRT. If anyone can answer, please go for it. Otherwise, I'll tell you the answer. Okay, I'll tell you the answer for it. Come on. Faster. Can you answer? If you can't answer, the answer is going to be glycogen and oil. Again, oil is like lipid. Okay, so glycogen is how the fungi will store the reserve food material. It is not like starch or anything. Okay, it is not going to be starch. It is going to be glycogen and oil. Okay, glycogen and lipid or glycogen and oil. Okay, so that is why I wanted to add this for all of you. Which one of the following is not a criteria for classification of fungi? I told you, on basis of what did they do the classification? Yes, it's not lipid here, it's not starch also, it's glycogen and oil. Which one of the, mainly it's glycogen, okay? Which one of the following is not a criteria for the classification of fungi? Mode of nutrition, mode of spore formation, fruiting bodies, morphology of mycelium. Can you think an answer? What did I tell you? Excellent, Money. Very good. Mode of nutrition, okay? Mode of nutrition. Very good. We did not consider because we know all of them are heterotrophic, right? Now, the characteristic feature of mycoplasma. The characteristic feature of mycoplasma is that presence of cell wall, lack of cell membrane, smallest living cell without a cell wall, double-stranded DNA in the nucleus. Okay, presence of cell wall. This is for previous one you're answering. Okay, students, you don't have to spam. Just tell once. Just tell once. I will be able to see. Okay. Yes, quickly answer. Three, very good. Smallest living cell without a cell wall. Excellent, excellent, excellent. 1 second. Yeah, good job. This is the answer. Okay, all of you are able to remember. I'm so happy. Which kingdom contains organisms that are mostly saprophytic? Easy question. Which kingdom contains organisms that are mostly saprophytic? Protista, Monera, fungi, plantae. Answer. Yes, very good. I think you people are answering for the previous question. I hope all of you marked your attendance. Okay, quickly smash the like button because we teachers are taking a lot of effort to make sure that your portions are completed. So I want all of you to be very, very cooperative. Okay, fungi, right? Fungi. Good job. So you have done. Okay, that's it, students. Yes, so here I've given the summary of the entire chapter. So I want all of you to read the summary and obviously utilize my notes. Okay, that's why I've taken the time and drawn all of this because I don't want you people to waste time by drawing. Okay, so I have drawn everything possible for all of you. Okay, yes. Oh, this would have got deleted. Done. Okay, I'll be sharing both the notes together. So don't worry about the notes. And students, yes, I wanted to tell you people about the JP Nagar Center. So students, if you want any details about especially the ones who are in Bangalore, okay, if you want to go to the JP Nagar Center, this is the number. You can book a seat or you can book a demo and you can go there. This is the address of it. JEE, NEET and 8th and 10th foundation classes will be taken. Okay, thank you so much, expert. Yes, so students, do not fail to comment. Okay, as I always say, do not fail to comment because your one comment that takes you 10-20 seconds is the one that motivates us teachers to go by, move fast. Just imagine we are just standing in front of the camera and talking, and if you people don't respond, you people don't comment, like we do not know, like how will we know how you like what is your feeling, how do you feel about the class? Okay, so I want a very mutual, okay, symbiotic relationship where I am going to teach you the best content in the best way possible and you are giving me all the love that you have to show. Okay, yes, thank you so much, students. See you all. Okay, I hope sir will give you a 5 minutes break. Okay, hey Kaushik, hello. Okay, yeah, he's my previous year student. Okay, bye bye. So sir will give you a 5-10 minutes break and join back for physics class. Okay, I will see you most probably next on Wednesday. Okay, Wednesday if I'm not wrong, but I'll be starting 12th that time. I'll be again taking a botany chapter, sexual reproduction. Okay, sexual reproduction in flowering plants, which is an amazing chapter, so we will learn it beautifully. Okay, yes, thank you so much Hema, thank you so much motivational buddies. Okay, see you all.